Hello everyone, I'm Stepan Tabadebaksh and I'm so honored and excited to present you one of the articles published in Nature. Today we are going to discuss a specific bacterial species with its potential role in refining wastewater. Nowadays, wastewater produced by sugar industries is damaging ecosystems of the US, and many bacterial species have been researched to combat this challenge. Meanwhile, Bacillus B.Marmarensis attracted scientists' attention to its potential resistance to contamination. In order to memorize the name easily, I broke down the Marmarensis word in Persian. Mar means snake, Meren in Mashhadi accent means goes, and also sis means stylish. Now we can code the name into this paragraph. This superhero in a snake shape goes into wastewater to convert sugars into ethanol in a stylish way, meaning that it's adaptable with a high pH environment. Here, we are supposed to answer the following questions. Why was this species selected? And how can this species convert sugars into ethanol? So the first step was to explore Bacillus B.Marmarensis properties. This graph clearly demonstrates that in which pH limit location this superhero has the most specific growth rate. As you move right towards the x-axis, the concentration of protons logarithmically decreases. The highest specific growth rate coincides with the pH extent between 9 and 10.5. Besides, in graph B, the specific growth rate of 2 E. coli and B. subtilis in neutral pH doesn't outweigh that of B.Marmarensis in pH of 10. Also in graph C, B.Marmarensis has exceeded the other three extreme alkaliphiles in terms of cell mass productivity. Furthermore, an experiment relating to the resistance demonstration to competitors was performed. The performance of 4 case controls lined in blue verified the accuracy of the experiment. Following that, the species competition was evaluated in 2 types of intentional contamination systems. One with open air exposure lined in yellow, which stays minute contamination from foreign species, and the second with moist and mountain soil lined in red, which the dominance of other species was significant as the amount of pH decreases. To sum up the results of the bacterial species selection, all graphs shows a promising approach of B.Marmarensis in high pH environments and the reason why this species is the most appropriate ones. As you can see in the left illustration, normally there are several fermentation pathways in bacteria with different flux intensity depending on their environment. In this case, 2 bioengineering protocols were applied to boost ethanol flux. The first protocol introduces the knockdown of lactate dehydrogenase gene, leading to suspension of pyruvate conversion to lactate, and the second protocol suggests the optimization of ribosome binding sites, which causes the overexpression of alcohol dehydrogenase gene and an increase in ethanol yield as we are looking up to it. As can be seen in graph D, by raising the percentage of sugar and buffer constant and removing peptones, the amount of titered lactate drastically grew. Additionally, the amount of succinate and acetate marginally reduced. Relying on the graph results, the application of 2 previously mentioned protocols is emphasized. From dozen designed plasmids in graph A, those ones lined in orange with antisense RNA based on the first protocol showed roughly 3-fold increase comparing to those lacking antisense RNA treatment. In graph C, the application of plasmids in green and red illustrate remarkable selectivity for ethanol. According to the second protocol, the result of the competition of 2 plasmids in red and green determines the final plasmid selection in graph D in terms of the highest ribosome binding site strength. Some experiments were also performed to certify the approach of the plasmid in red. In graph E, by increasing the multifold amount of sugar, the ethanol titer increases. Besides, in graph F and G, the ethanol yield percent in unsterilized environment is about 10% less than that of in a sterilized environment. A glance at the graph G states 54% ethanol yield from the final bioengineered B.Marmarensis. Comparing to other species in extreme conditions, including high amount of pH and competitors' involvement, our bioengineered superhero may show a substantial effects in biorefining of sugar-contained contaminations in wastewater of sugar factories. At the end, I want to thank you all for paying attention to my presentation, and especially Dr. Zamani and Dr. Leah for holding this beneficial science communication course. If you have any questions, I'm honored to answer.